Chapter 2 Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for our own use for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' times, a two-kingdom system of classification with Plantae and Animalia kingdom was developed that include all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic and non-photosynthetic organism. Classification of organism into plants and animal was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of fall, mode of nutrition, habitat, method of reproduction, evolutionary relationship, etc. Classification system for the living organism have hence undergone several changes over the time. Thought, plant and animal kingdom have been a constant under all different systems. The understanding what group or organism be included under this kingdom have been changing. The number and nature of other kingdom have also been understood differently by different scientists over the time. Table 2.1 Characteristics of five kingdoms. Character, five kingdoms. Character, cell type Monera, prokaryotic, cell wall Monera, non-cellulosic in bracket polysaccharide plus amino acid, nuclear membrane Monera absent, body organization Monera cellular, mode of nutrition Monera autotrophic in bracket chemosynthetic and photosynthetic bracket close, and heterotrophic in bracket saprophytic or parasitic bracket close. Protista, cell wall, cell type eukaryotic, cell wall present in some, nuclear membrane in Protista present, body organization Protista cellular, mode of nutrition Protista autotrophic in bracket photosynthetic and heterotrophic. Cell type fungi eukaryotic, cell wall fungi present with chitin, nuclear membrane fungi present, body organization fungi multicellular or loose tissue, mode of nutrition fungi heterotrophic in bracket saprophytic parasitic. Plantae, cell type eukaryote, cell wall present in bracket cellulose, nuclear membrane plantain present, body organization Plantae tissue or organ, mode of nutrition Plantae autotrophic in bracket photosynthetic. Animalia, cell type eukaryotic, cell wall Animalia absent, nuclear membrane Animalia present, body organization Animalia tissue or organ system, mode of nutrition Animalia heterotrophic in bracket holozoic or saprophytic, etc. Bracket close. R.H. Whitaker 1969 proposed a five kingdom classification. The kingdom defined by him were name Monera, Protista, fungi, Plantae and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationship. Table 2.1 give a comparative account of different characteristics of the five kingdoms. The three domain system has also been proposed that divide the kingdom Monera into two domains, leaving the remaining eukaryotic kingdom in the third domain and thereby a 6 kingdom classification. You will learn about this system in detail at a higher class. Let us look 5 kingdom classification to understand the issue and consideration that influence the classification system. Earlier classification system including bacteria, blue-green algae, fungi, mosses, fern, gymnosperm and the angiosperm under plant. The character that unified this whole kingdom was that all the organisms include had a cell wall in their cell. The places together group which widely different in other characters. It brought together the prokaryotic bacteria and the blue-green algae in bracket cyanobacteria with other groups which were eukaryotics. It also grouped together the unicellular organism and the multicellular ones. Say for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants. True, they also show a characteristic difference in their walls composition. The fungi had chitin in their wall, while the green plant had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom. Kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, Chlorella, in bracket earlier placed in algae within plants and both having cell wall, with Paramecium and Amoeba in bracket which were earlier placed in the animal kingdom which lack cell wall bracket close. It has put together organisms which in earlier classification were placed in different kingdom. This happened because the criteria for the classification change. This kind of change will take place in future to depending on the improvement in our understanding of character and evolutionary relationship over time. An attempt has been made to evolve a classification system which reflect not only the morphological, physiological and reproductive similarity but is also phylogenetic, is based on the evolutionary relationship. In this chapter, we will study characteristics of kingdom Monera, Protista and fungi of Whitaker's system of classification. The kingdom Plantae and Animalia, commonly referred to as plant and animal kingdom respectively, will be dealt separately in chapter 3 and 4.